a new type of structure we have developed for biomedical applications. We call it the nanodomes. This work is a collaboration between my group at the Catalan Institute of Nanoscience and Nanotechnology, the Universitat Autònoma de Barcelona, and the SIC Nanogune in San Sebastián. So what is a nanodome? What we call a nanodome is we have a polystyrene sphere, usually of 100 nanometers, where we grow semi-shells on top. And we grow two types of shells. First, an iron shell, which is magnetic, and then on top of that, a gold shell, which is plasmonic. And with this structure, we want to do optical hyperthermia for cancer treatment. So what's the role of the gold layer? We have the gold has a localized surface plasmon, which we can use for optical heating. Then it also has a high atomic number, which can be used for X-ray computer tomography contrast. And it's very easy to biofunctionalize the iron layer. What's the magnetic layer giving us? Iron, since we use iron, it has a high magnetization. It also has a strong nuclear magnetoresonance response, which can be used as MRI contrast. And it's also, since it has a high magnetization, it's very easy to manipulate it using magnetic fields. So how do we go about this? So first we get commercial polystyrene spheres. We usually use 100 nanometers, but you can use other sizes. And then we self-assemble them on a silicon wafer. And then we evaporate the iron and the gold layers using electron beam evaporation. And then if needed, we can functionalize them while they are still on the wafer. And then using ultrasonication, we can transfer them to water. I show you here a SEM picture of the self-assembly of the polystyrene spheres and a TM picture of the final nanodomes. Actually, using this 4-inch wafer, we create about 110 to 11 particles per wafer, which translate into 600 micrograms per wafer, and is easily scalable to higher wafers. So what's the advantage of this approach is we do with this synthesis in vacuum, so we don't need any toxic organic solvents. It allows us to directly functionalize the nanoparticles while they're still on the wafer. It's very easy to disperse them in water, and they are highly colloidal. They have a high colloidal stability, and they have a tunable magnetic and optical properties. I'll show you now. So we measure the magnetic properties by changing the ratio of iron to gold. For example, here we see we have 5 nanometers of iron and 35 nanometers in gold. The total thickness is always 40 nanometers. So, and then we see that when we have 5 or 10 nanometers of iron, we see a single domain behavior. So meaning that all the moments point in the same direction, like this. However, as we increase the thickness of the iron layer, the system no longer has a single domain behavior, but has what is called a vortex, magnetic vortex behavior, where the magnetization curls around the spherical shape of the nanodome. This is very important because this, as we can see here, this has zero remanence at zero applied field. This means that this system has very weak dipolar interactions, which avoids agglomeration. And this translates into high colloidal stability. And as I said before, it's very easy to manipulate them using magnetic fields. As a simple note, one nanodome is equivalent to 500 superparamagnetic iron oxide nanoparticles. That means that it's much easier to manipulate the nanodomes than the superparamagnetic iron nanoparticles because they have a much higher total magnetization in the system. As for the optical properties, we can see here This is in the pure gold has a nice plasmonic resonance at about 800 nanometers. Importantly, this wavelength is in the first biological window, so where the light penetrates the most inside the body. Also, as we start adding iron into the system, first there is a redshift of the plasmonic resonance, and there is also a damping of the plasmonic resonance. As I said, this can be used for heating. The way we go about this is we have a suspension of nanoparticles inside the cuvette. We apply 808 laser, and then using an infrared thermometer, we measure the temperature of the liquid. We see here that when we apply the laser, here in blue is the intensity measured at the power meter, the temperature of the water increases, and in about 10 minutes we can get an increase of temperature in the water about 20 degrees. Also, the heating ability of the particles depends on the concentration, as expected, and we see that for higher concentration it tends to saturate. This is because at higher concentration the nanoparticles absorb so much light that the light does not reach the last particles. So eventually only the first part of the particles heat, so that's why there is a saturation. And also interestingly, the heating properties are quite independent of the iron content. Also, in terms of the imaging of these nanoparticles, first, as I said, these particles have a strong nuclear magnetic resonance. Here I put you the example of the layer with 30 nanometers of iron, which has a T2 of 266 millimolar per second. As a comparison, the commercial system based on iron oxide, it has 176, so we're doing much better. And here I show you an example of a real experiment. This is as we just injected the particles in the liver, and we see here in gray, and after a few hours we see that this becomes black. It means that we have a very high contrast on the areas where we deposit the nanoparticles. Also, these nanoparticles are very efficient in absorbing X-rays. Here I show you an X-ray microscopy image of nanoparticles inside frozen cells. Here you see the organelles of the cells, and here in black are the nanodomes, which are highly contrasting black, meaning they absorb a lot of the X-ray energy. And this is interesting for computed tomography, although we have not tried it yet. And another imaging capability is that nowadays you can buy fluorescent polystyrene beads. So now you can image these beads using confocal microscopy. Here I show you some confocal microscopy images of cells. This is a nucleus, with the red dots here inside are the nanodomes. And here in the computed tomography we can do a 3D scan, and here I showed you the projection of these cells, and you see that indeed the nanoparticles are absorbed inside the cells. So as I show you, the iron layer allows us for easy manipulation. That's done using what's called a magnetophoretic effect, where when we put the magnetic field, the particles tend to go towards the magnetic field. So here I show you how we can control the heating using the magnetic field. Here, in this case, the laser is pointing towards the screen, and we have the magnet on the side. So when we put the magnet, what we basically do is to remove the nanoparticles from the laser path. And what this does, that when we apply the laser without magnet, it starts heating. And now we apply the magnet, the particles go away from the laser path, so then they don't heat anymore. And here you see here the transmission increases very much. But we can do the other way around. Now the laser is going this way. So now with the magnet, we can concentrate the particles in the laser path. And here what it does when we first apply the laser, it starts heating as before. And now when we concentrate the particles in the laser path, it increases much more efficiently. So we can enhance the magnetic heating just by using the magnet, by concentrating the particles in the path of the laser. So now we try this idea in vitro. So we incubated HeLa cancer cells, and we use nanodomes of 20 iron, 20 gold, and we use an 810 nanometers laser at 5 watts per square centimeters for 30 minutes. So then we first applied a magnetic field to see what happens. Then we applied only light to see what happens. And then we combined it with nanoparticles. You can see here that magnetic field and laser that do nothing to the cell viability, and magnetic field with 10 micrograms of nanoparticles do nothing. Magnetic field with 100 micrograms of nanoparticles, still nothing. Light with 10 micrograms do nothing. Light with 100 micrograms starts killing some cells. And then when we do light and magnetic field with 10 micrograms, we already see some decrease. That's after 24 hours. But if we wait for 48 hours, we see that most of the cells are killed by this process. Similarly, if we do 100 micrograms with light and magnetic field for 24 hours, we also kill most of the cells. So we see here the optical micrograph of the cells here in green when they are alive, in red when we kill them. But then we did an SEM of the cells. We see before treatment they are nicely spread on the Petri dish, and when we use only 10 micrograms, you see that the cells have deformed, but they still look like cells. Meaning that here the cells are killed by apoptosis. However, when we do the treatment with 100 micrograms, we see that the cells are completely destroyed, like burned out. And meaning that in this case, we kill the cells by necrosis. This kind of cell killing is not appropriate because it leads to side effects. So this brings us about what is the actual temperature in the liquid surrounding the particle. We need to do that to be able to tune the conditions for the optimal killing of the cells using only apoptosis and not necrosis. So then it brought us, can we measure locally the temperature of the nanoparticles, all the liquids surrounding the nanoparticles? To do that, we design a new type of nanoparticle, a nanodome. Instead of having gold and iron, in this case we use cobalt and iron multilayers capped with some gold. Here I show you a TM picture of the image, and here it's a energy loss spectroscopy, when we can see that we do have a multilayer of gold and cobalt. And this, in contrast to the other particles where they had an in-plane magnetization pointing this way, now in this multilayer, the magnetization is out of plane. This gives the nanoparticles a much stronger magnetic anisotropy. So what we did next is to disperse the nanoparticles inside the liquid. Again, put our laser light, 800 nanometers. But now we have a magnetic coil here. And now we measure the transmission. And you see here that the transmission changes very much when we apply magnetic fields, especially at 800 800 nanometers. Why is this? Because at zero field, the nanoparticles are randomly distributed, but when we apply the field, all the particles align to the field, and then the absorption, since these particles have an asymmetric optical properties, when they are random, the absorption is very different than when they are aligned. That's why the transmission changes very much with the applied field. So now we use this idea for sensing. So we do a dynamic magnetochromic effect due to the particle rotation. So what we do is to shine light, and then we put an AC field in the coils. And there is two effects in the transmission of the light. You see here in blue is this is the applied magnetic field, and in red is the response. The first thing you can see right away is that the response of the nanoparticles is twice the frequency of the applied magnetic fields. This is because optically the up and down are equivalent. So the response in the transmission, it has a double the frequency. And also there is a very important effect shown here by this line, is that there is a phase lag between the applied field and the transmission response. And this is due to the viscosity of the liquid, meaning that the more viscous the liquid, more difficult it is for the nanoparticles to rotate. So since the viscosity of a liquid depends strongly on the temperature, we will use this effect to measure the temperature surrounding the nanoparticles. So what we did is we applied heating using a Peltier, and then we check with our infrared thermometer the temperature of the liquid, this here in black. So as we apply the Peltier, the temperature of the liquid increases. And then at the same time, we measure the transmission of the suspension. And here I show you the phase lag at the same time as we heat. And you see that there's a very nice correspondence between the temperature of the liquid and the phase lag of the system. So we can use this to calibrate our liquid, see how much phase lag each degree brings about. So actually, using this approach, we can do real-time detection of temperature variations. And actually, we have a limit of detection of about 0.05 degrees. So now, our idea was, what about if we, instead of a Peltier, we use the actual laser to hit the nanoparticles? Can we still measure? Indeed. Here I show you that when we put the high power on of the laser, the temperature measured with the infrared thermometer increases, and the temperature derived from the phase lag also increases, and they nicely follow each other. Actually, there is a slight difference in temperature between the infrared and the nanodomes and the phase lag. This is because the infrared only measures the surface temperature, and it takes actually some time for the temperature to diffuse the whole liquid, while the nanodomes measure the temperature inside the liquid surrounding the nanoparticles. So also this idea allows us to fast detect the temperature. What we tried is to, instead of having a continuous laser power as before, now we use pulses of power of a few seconds. And you see that infrared thermometer cannot follow the fast changes, and it only sees after a few minutes, it sees the overall effect of these pulses. But with nanodomes, we can actually follow the temperature changes inside the liquid as we pulse a laser. So the response of the nanodomes is much faster than the infrared thermometer. So to conclude, I hope I have convinced you that we can use iron gold nanodomes to control photothermal therapies, These are strong nanomagnets and strong nanoheaters. We can do multimodal imaging using the nanodomes, both NMR, computed tomography, and fluorescence. And they have a very high efficiency photothermal therapy enhanced by magnetic manipulation. Using the cobalt gold nanodomes, and actually we can do something similar with the gold iron, which is a little bit less effective than the cobalt gold. We can use them as nanoheaters and simultaneous nanoheaters and nanothermometers. Because they have a high magnetic and optical anisotropies, which we use for rotation of the nanoparticles and for the sensing. We can detect the viscosity and temperature variations with high sensitivity. And we can simultaneously efficiently optical heat and sensitively detect the temperature even in cell dispersions. So thank you for your attention.